session of the chapter number 4, which is the cell membranes and transport. So here basically, as this is chapter number 4 of your A-level AS biology 970 and 220. Now we are starting from the 4.1 fluid mosaic membranes. And wait a minute. Alright, so in the fluid mosaic membrane, what this is basically required to just understand is that you can just read from this is that in the fluid mosaic model, you can just read that the fluid refers to the movement of phospholipids, while the mosaic refers to the scattered proteins and glycoproteins in the phospholipid bilayer. So we are just required to just understand about the structure of the phospholipids. So you can just see as this diagram is very clear to you, I hope so, that there is a hydrophilic head and a hydrophobic tail. What does the hydrophilic and the term hydrophobic means is that the hydrophilic is the water loving and the hydrophobic means the phobia of water, means water hating. Okay. So if meanwhile that head is attached to the water molecules and the tail is not attached to the water molecules and it also repels because it is hydrophobic or the water heating molecules. So these okay, as you can see that there's a fatty acids are being present in the hydrophobic and they are the phosphate and glycerol present in the hydrophilic heads. Whenever the glycogen molecule or the glucose molecule is attached to the lipids or the proteins, so they are called as glycoproteins and glycolipids. All right, so this will be understanding much more. Phospholipids are arranged so that hydrophobic non-polar tails do not face water. Non-polar means that they are insoluble in water. Understand? Water is on both the intracellular and extracellular sides. Meanwhile, inner side and outer side of the cell. Okay. Therefore, tails point inwards and hydrophilic heads face the aqueous medium. As you can see that this is visible to you, and in this diagram you can just see clearly that the heads are facing towards the water. It means that there are polar heads and the non-polar which are not soluble in water and are water hating as hydrophobic. So they are, as you can see, this is called bilayer. Okay. So this is the diagram you are required to just keep in your mind, and this is called as lipid bilayer. What is membrane fluidity? Membrane fluidity refers to the viscosity of the lipid bilayer of a cell membrane. It only depends on the viscosity of lipid bilayer molecules. Okay. Now wait a minute. Now, okay, so now what is basically is membrane fluidity is affected by two things. As you can see, okay, not two things. As you can see from this tail length, the tail length is very essential for the membrane fluidity as it affects by the longer the tail, the less fluid the membrane. All right, if the tail is long, so you can see that there's less fluid. Okay, the saturation of fatty acids in the saturation of fatty in the saturation. Wait a minute. Okay, so we were at the membrane fluidity affected by the tail length as the tail length. Okay, so the longer the tail, the less the fluid membrane. Okay, so now we are just required that we are required to just make the relationship. Tail is inversely proportional to membrane fluid of membrane or the membrane fluidity. Okay, now the saturation of fatty acids. What does the saturation of fatty acid means? It means basically that the more unsaturated they are, the more fluid the membrane. Okay, this is as unsaturated fatty acids tails are bent and fit toward together more loosely. Okay, and the cholesterol regulates the fluidity of membrane. Plus, at low temperatures, cholesterol increases the fluidity of the membrane, preventing it from being too rigid. This is because it prevents coarse packing of phospholipid tails. At higher temperatures, cholesterol decreases the fluidity of membrane and stabilizes the cell. It is not talking about in the journal. I know when you'll be understanding temperature relationship, so you can just think that how is it so possible as it is vice versa, and this is the relationship of the directly proportionality. As the temperature increases, so the fluidity must increase, and when the temperature decreases, so fluidity must decrease. But how it has been happening, so you have not focused that when temperature increases, the fluidity is decreasing by the cholesterol. All right, so this is the factor number three we are describing. So this is the cholesterol. When the temperature increases or decreases mean of cholesterol. Okay, yeah, but in the journal, if the temperature increases, so the fluidity must increase. Number two and number three are combined as glycolipids and glycines. Okay, so whenever there is the fluidity, whenever the glucose or glycogen molecules are attached to lipid molecules or protein molecules. Okay, so they are called as glycoproteins and glycolipids. All right, and the out. Okay, so as you can see that lipid and protein molecules on the outer surfaces of cell membrane have carbohydrate chain attached to them, forming glycoproteins and glycolipids. These carbohydrate chains projecting out like antenna. As you can just, I will just make you understand from the example of the diagram that it stabilize the membrane structure by forming hydrogen bonds with water molecules surrounding the cell. And there's a glyco, a sugary cell coating formed by carbohydrate. They act as receptor molecules, signaling receptors, and endocytosis. We have not already discussed endocytosis in our live stream of the cell structure, where the endocytosis means to bind to molecule to be engulfed by a membrane. Okay, and the cell signaling means signaling receptors as recognize messenger molecules like hormones and neurotransmitters. All right. As for suppose that whenever you get a pain in your hand or in your muscles, so there's a rapid reaction. This is because of the neurotransmitters which have process. Okay, and they are called as signaling receptors and act as cell markers and antigens, allowing cell to cell recognitions. Moving towards okay, so there are plasma proteins which are of two types, one integral and the peripheral. We are talking about the integral means a transmembrane protein. So we were been talking about the transmembrane protein. So these are called the proteins that are found embedded within the membrane, may be found in inner layer, outer layer, or spanning the whole membrane. These are transmembrane. Help in movement and in and out of cell. What does it mean? Diffusion, facilitated diffusion, osmosis, active transport, and bulk active transport. These are the five things you are just required to just learn in this live session. All right. And moving towards the peripheral. Okay. The peripheral plasma proteins can be present inside of the cell membrane, example intracellular and extracellular. Intracellular peripheral proteins are the communication receptors and recognition proteins. Alright, and intracellular peripheral proteins are structural support attached to the cytoskeleton of the cell. And now moving towards the function of transmembrane proteins. So remember that there are two types of protein, transmembrane proteins. They act as gateways and can be transformed, helping in facilitated diffusion and active transport. This will be understanding, okay. As you can see from the diagram, the phospholipid is been visible to you, the glycoprotein is visible to you, and the phosphate head and the okay, so the phospholipid is also visible to you. As you can see that the fatty acid tails are being given to you, okay. The carrier protein and channel protein, these both are the intracellular proteins or integral proteins. All right, what is channel protein? Basically, the channel proteins do not require energy and they transport substances through membrane along their concentration gradient, used for both active transport and facilitated diffusion. Why it does not require energy? This is because they work along their concentration gradient. If the concentration is moving from higher to lower, they'll be moving from higher to lower. If the concentration is from lower to higher, it will be moving from lower to higher. They'll be moving across along their concentration gradient, so it does not require any energy, and they transport the substances through membrane passively. All right. Whereas the carrier proteins are requiring energy, this is because they go against the concentration gradient, which requires energy from ATP, obviously, and they take substances from outside and pumps it inside, or vice versa. Means outside to inside or inside to outside, and used for active transport, which requires ATP. Cell surface receptor particular substances used for signaling endocytosis, cell adhesion, cell markers. Cell surface antigens acts as cells identifying markers. Each type of cell has its own antigen. This enables cell to recognize other cells and behave in an organized way. What does cell signaling means? The cells detect signals with cell receptors such as glycoproteins and glycolipids present on their membrane. These are the cell receptors. The signaling molecule binds to the receptor as the shapes are complementary to the each other. As we have already discussed in the enzyme live video that their enzyme is having the complementary shape towards the substrate, so that substrate could easily bind towards the active site or region enzyme. So complementary means if one of the enzyme is having each type of shape, so the substrate must have the shape which could be attached perfectly towards enzyme. So this is called complementary. Okay, and this creates a chain of reactions in the cell leading to a response. All right. Okay, if the signaling molecules are hydrophobic, such as steroid hormones such as estrogen, they can diffuse directly across the cell membrane and bind to receptors in the cytoplasm or nucleus, as this is visible to you. Okay, if the signaling molecule is water soluble, number one, signal arrives at protein receptor in cell membrane. Right, the receptor's shape is complementary to the ligand. What does ligand means is basically a molecule which is from foreign, okay, which is not of that particular cell. The signal brings about a change in the receptor's shape. Changing the shape of the receptor allows it to interact with the next component of the pathway, so the message gets transmitted. Binding triggers stimulates reactions within the cell. Cell signaling results in a response which may be intracellular or extracellular. So as you can see, as the cell signaling results in a response which may intracellular or extracellular. Now you can here be seen very visible to you that there is the extracellular fluid and there is a cell. As number one is reception, when the receptors and the signal molecule, which is the ligand, is being joined towards this what type of molecule? The transduction, the signal transduction pathway, as you can see now, the shapes are being changed, and at the response, the activation of cellular responses here. Okay. Number four, and this is now finally we are moving towards the last step and discussing the five things as diffusion, facilitated diffusion, osmosis, active transport, and bulk active transport. What is new for you guys is bulk transport and facilitated diffusion from your O levels to A levels. Okay, what is diffusion? Net movement of molecules or ions from a region of higher concentration to a region of lower concentration down a concentration gradient. As a result of the random movement of particles, passive process, molecules tend to reach an equilibrium situation. And as you know, the diffusion occurs and occurs until and unless they have an equilibrium observed. Okay, factors affecting diffusions are the as the steepness of gradient increases, diffusion increases; as temperature increases, diffusion increases; as surface area increases, diffusion increases; distance increases, diffusion decreases. And smaller and non-polar molecules like fats diffuse much easily across the cell surface membrane as they are soluble in phospholipid tails. Now, what is facilitated diffusion? Diffusion of a substance through transport proteins in a cell surface membrane means now it is requiring a particular molecule or particular protein, which is called transport protein. It means that the diffusion is occurring by facilitating it. It requires a facilitate. Okay. The proteins provide hydrophilic areas that allow the molecules or ions to pass through the membrane which would or otherwise be less permeable to them. As the channel proteins, you can see that carrier proteins and channel proteins. These are the both these are the both right. So there have been two types of functions. Okay, there are two types of protein. One is channel protein and one is carrier proteins, which are also called as transport proteins. Means they are being function in the facilitated diffusion also, as the channel proteins allow charged substances, usually ions, to diffuse and can move to open or close the pore like a gate controlling ion exchange. All right, as you can see that this is a is working as a simple pathway at the intracellular space, and you can just see what has been happening. All right, I hope so that these diagrams are clarifying you guys. Okay, and the carrier proteins are basically flipped between two shapes. As a result, the binding site opens alternatively to each other. All right, what is osmosis? The net movement of water molecules from a region of higher water potential to a region of lower water potential through a partially permeable membrane as a result of their random motion, and this is throughout the concentration gradient. Okay, water potential, the tendency of water to move out of solution. Water always move down a water potential gradient. This happens until water potential is the same throughout the solution, and it is denoted by this symbol, as you can see. Okay, and water potential becomes negative if the solute concentration is very high. Now the water loss and water gain in the animal cells and plant cells. In the animal cells, or RBCs means red blood cells. Okay, when the water is lost from the animal cell, it becomes crenated, and when the water is lost from plant cells, they plasmolyzed or flaccid. Okay, when the water is gained by animal cells, so it is hemolyzed or symbolized. Okay, and in the plant cell, it turgid. Okay, moving towards the active transport, movement of molecules of movement of molecules or ions through transport proteins across the cell membrane against their concentration gradient using energy from ATP. Now, do you understand why the active transport requires energy from ATP? This is because that they are working against the concentration gradient, okay? And it is achieved by carrier and channel proteins. These are specific to the type of proteins they are transporting, and the requires energy supplied by ATP, means adenosine triphosphate. Energy is used to make the channel carrier change shape, transferring molecules, ions across the membrane in the process. Now, understanding the sodium potassium pump. All right. So, understanding the sodium potassium pump, as you can see that in the sodium and potassium pump from this figure, the extracellular fluid and the cytoplasm are being visible to you. Cytoplasm means intracellular fluid, okay? So the sodium potassium exchange pump means that the ATP, okay? Yes, the three molecules of sodium moves out, and the two molecules of calcium, or sorry, potassium moves in. Okay, from extracellular fluid, the two molecules of potassium enters the cytoplasm, and three molecules of sodium moves out of the cell membrane. All right, this is the cell membrane. As you can see that these red balls are the hydrophilic heads, and the hydrophilic tails are also visible to you. This is the cell membrane. Okay, so this is sodium potassium pump. For every ATP molecule used, 3 sodium is given out of cell and 2 potassium taken in the cell. This is what you're required to just learn. And then the last, moving towards the bulk transport, a type of active transport where large molecules are transported across the cell surface membrane using energy from ATP. There's the endocytosis and exocytosis. Just wait a minute, it could be very clear to you guys. In the endocytosis, as we have already discussed, that this is an engulfing of the molecules, okay? And exocytosis, okay. See that endocytosis means from inside and exocytosis means outside. We have discussed in the first in the live session of the chapter number 1. Now you can see from this figure very easily that what has been happening, that whenever the exocytosis occurs, so the exocytosis vesicle is okay leaving the cytoplasm. As you can see that how it is leaving and how it is entering, okay, or forming. When it is leaving, it is called endocytosis, and when it is entering, it is called when it is leaving, it is called exocytosis, and when it is entering, it is called endocytosis. Endocytosis, bulk movement of liquids means pinocytosis, or solids, phagocytosis into a cell by the enfolding of a cell membrane to form vesicles containing the substance. What is pinocytosis and phagocytosis? It is very simple. I'm giving to you is that whenever you drink, this is called pinocytosis. Okay, and whenever you eat, so you it is at phagocytosis. As you can see, the solids are phagocytosis and the liquids are pinocytosis. Moving towards the last, exocytosis, bulk movement of liquids or solids out of a cell by the fusion of vesicles containing the substance with the cell surface membrane. So I hope so that this is the video and you have understand everything from this video, and I hope so that this is clarifying very easiest chapter in the your biology. And if there is a difficulty, just wait for the comments and just comment me below and wait for the reply. Until that, if you have not subscribed the channel, so then subscribe the channel. If you are new to channel, just visit the channel. If you love it, so just give the good feedbacks to in the return. Until take care.